About the claims that tylenol during pregnancy causes autism? Well, to evaluate that claim, we must understand that neurodevelopmental disorders such as autism are extremely complex, and at the moment there is no convincing evidence for Tylenol being a direct cause of autism development in children. Both the World Health Organization, WHO, and U.S. Food and Drug Administration, FDA, have confirmed Tylenol as the safest over-the-counter pain and fever medication. So that begs the question, how are neurodevelopmental disorders caused? Well, to answer that, let's dive into understanding neural development. Neural development is the process by which the nervous system is generated, patterned, and refined. It starts early in embryogenesis and extends into the postnatal period. It starts with neurogenesis, where neural stem cells lead to three major neural cell types. These are the neurons, astrocytes, and oligodendrocytes. The majority of neurogenesis happens prenatally, as it is when rapid cell proliferation establishes the foundational architecture of the brain. However, there is still neurogenesis in adult periods. This persists in specific regions, specifically in the subventricular zone, which contributes new interneurons, and also the dentate gyrus of the hippocampus, which has newly generated neurons that are involved in learning and memory. Once the neurons are generated, they reach the appropriate location through neuronal migration. Most of the cortical neurons come from the ventricular zone and migrate to the cortical plate, which is a more outward structure. This migration is guided by radial glial scaffolds. These are both the progenitor cells and are also structural guides. The disruption of migrations can lead to severe neurodevelopmental disorders. For example, in autism, disrupted neuronal migration subtly alters cortical layering and regional connectivity rather than causing gross malformations. These early migration differences can bias how neural circuits are wired, which contributes to the atypical connectivity and information processing that we usually see later in development. After the migration, neurons establish long-range connections through axon guidance. The axonal growth cone navigates the extracellular environment because it is able to detect molecular guidance cues, which includes netrins, semaphorins, ephrins, and slits. These cues work through chemoattraction and chemorepulsion, which makes sure that the axons extend toward correct targets and still avoid inappropriate regions. Precise axon guidance is important in terms of forming functional neural circuits. Next, the neurons make synaptic connections through what is called synaptogenesis. During this stage, neurons make a lot of synapses, which is often more than what will be retained in adulthood. Synaptogenesis is affected by activity-dependent mechanisms, meaning neural activity makes appropriate connections stronger. Astrocytic signaling also plays an important role by releasing factors that promote synapse formation and stabilization. Excess synapses are later eliminated through synaptic pruning. This makes sure that neural circuits are efficient. At the same time, neural differentiation starts to pattern a developing brain. This means that the neural tube initially forms three primary vesicles, which is the prosencephalon, mesencephalon, and rhombencephalon. Those then end up subdividing more into secondary vesicles, which include the telencephalon, diencephalon, metencephalon, and myelencephalon. Ultimately, these give rise to distinct brain regions. Finally, neural development is affected by critical periods, which are time-restricted windows of more neural plasticity. During these periods, experience is very important in refining the circuits. This is why sensory deprivation, for example, during a critical period, can lead to permanent functional deficits. The opening and closing of critical periods are dictated by inhibitory circuit maturation, myelination, and the formation of perineuronal nets. These stabilize synaptic connections and limit further plasticity. Together, these processes make sure that cells assemble and mature in the nervous system in a perfect way. And when a part of this process goes wrong, it gives rise to diseases such as autism and other neurodevelopmental disorders.